Sathyabama Institute of Science and Technology.
A very good day. This is Preetha Wilma Dawson, Assistant Professor, College of Physiotherapy, Sathyabama Institute of Science and Technology, deemed to be a university. In today's lecture, we'll be starting with the structure and function of neuron. The nervous system is basically composed of two distinct cells. The first one is going to be the neurons and the other one is going to be the neuroglial cells. So first, we'll get started with the neuron. So neuron is a basic structural and functional unit of the nervous system. It is a true functional cell in the nervous system and it is a form of specialized cell which is working in the nervous system. And these neurons are conductible cells, that is, they have the ability to transfer the action potentials. Next, we'll get started with the neuroglial cells. Glial. The word glial is derived from the Greek word with the meaning glue. So, this neuroglial cells is also known as glial cells or glia. So, these cells are not the true functional cells like neurons, rather they are the supporting cells present in the nervous system. And the number of glial cells are more when compared to the number of neurons in the nervous system. But the size of the glial cell are small when compared to the neuron size. These cells are non-conductible cells, that is, they don't have the ability to transfer the action potential. Now, let's discuss about the structure of neuron. So, this is the structure of neuron. We have a cell body which is also known as soma. In between the cell body, we have a nucleus which is surrounded by an extensive tapering structure which is called as dendrite. Again, we have a tubular-like structure which is called as axon, and the structure which is called as axon hillock is a structure which is connecting the soma, that is the cell body, and the axon, which is a tubular structure. This interconnecting structure is called as axon hillock. And we have the myelin sheath which is covering the axon. So, in between the myelin sheath, we have some specific nodes which is called as node of Ranvier. And again, on the myelin sheath, we have a special type of cell which is called as Schwann cells. And this axon will have a finger-like projection at the end which is called as axon terminal. So, let's discuss about each structure individually. To get started, we'll start with the dendrite. So, this dendrite is going to be the afferent component, that is the sensory component of the neuron. So, they will be branching extensively into a dendritic tree and they will be tapering distally. As they are tapering distally, they are going to reduce in the diameter and they are the conduits for receiving neural signals. The dendrites are very thin processes which will be extending from the soma, which is also known as cell body, and this will be serving as a primary receiving end or primary afferent end or primary sensory end. Over 90% of synapses in the neurons are located within the dendrite. So, this is a structure of dendrite. Here you can see the dendritic tree which is tapering distally. Next, we'll start with cell body. As I have said before, the cell body is also referred as soma. So, cell body is sometimes also called as neuron cell body. It is the main part of the neuron which consists of nucleus and also other organelles also present like Golgi apparatus, mitochondria, endoplasmic reticulum and other essential organelles are also present within the soma along with the nucleus. Next, this soma will play a very crucial role in both processing and also transmitting the information within the nervous system. So, inside the soma, we have a special type of organelle which is called as Nissl body. And this Nissl body is very much essential for the synthesis of protein. And they are also very crucial in the regeneration of any injured axon. Let us consider if there is going to be any fatigue or injury of a neurons, what will happen is this Nissl body will fragment and disappear. So, this fragmentation and disappearing will occur by the process of chromatolysis, and we'll start with the nucleus. So, each neuron will be having only one nucleus and the nucleus in this neuron will not contain a centrosome. Because of that, the nerve cell does not have the ability to multiply. This is going to be the picture representation about the soma. So, you can see here we have a nuclei surrounded by the other essential organelles in the soma. Next, we'll start with axon hillock. It is a specialized part of neuron that will separate the cell body from the axon, as I have discussed before. So this axon hillock is a triangular cone-shaped structure which will separate the cell body, that is the soma, from the axon. It is going to be a triangular structure in a conical shape which will be continuing as an axon. The axon hillock plays a very important role in the neural communication. So here you can see this is the cell body which is extending into the axon, and here you have a triangle conical structure which is called the axon hillock, which is separating the cell body from the axon. Next we will start with the axon. So axon is the longer process of the nerve cell. Each neuron will have only one axon. The axon, as we discussed before, the axon will be rising from the axon hillock of nerve cell body, and it is devoid of nucleus granules. Yes, while talking about the soma, we discussed that they have a specialized organelle which is called as nucleus body, and this nucleus body is responsible for protein synthesis. But this axon is completely devoid of nucleus granules, that is, they do not possess nucleus granule inside them. So the length of the longest axon is of about 1 meter. So the axon will be transmitting impulses away from the nerve cell body to the axon terminal. So each axon will be having a central core of cytoplasm. So what is cytoplasm? A cytoplasm is nothing but a gelatinous liquid which is present inside the cell. So it is mainly responsible to maintain the contour of the cell. So this axon will have a central core of cytoplasm. So the central core of cytoplasm in the axon is called as axoplasm. So this axoplasm will be covered by a tubular sheath which is again called as axolemma. So axoplasm is going to be the central core of cytoplasm, and the cytoplasm layer will be covered by a tubular sheath which is called as axolemma. And again this axoplasm and the axolemma together is called as axis cylinder. So again axis cylinder is surrounded by a layer which is called as neurolemma. And then we'll start with axonal flow. As I have discussed in the previous slide, so this axon is completely devoid of nucleus granule. There is a specific requirement of the protein synthesis in the axon for proper nerve functioning, but the axon is completely devoid of nucleus granules, so there is going to be no protein synthesis. So what will happen is the protein which is synthesized in the soma, that is the cell body, will be transmitted from the soma to the axon, and this means of transmission is called as axonal flow. Here you can see this is the axon which is getting connected to the axon terminal and which is covered by a layer of sheath which is called as myelin sheath. So now let's get started with a myelin sheath. A myelin sheath is a thick lipoprotein layer that insulates a myelinated nerve fiber. If a fiber is going to be a myelinated fiber, it will possess myelin sheath. If a fiber is a non-myelinated, it will not have a myelin sheath insulating over it. So this myelin sheath is a lipoprotein layer which is formed by a concentric layer of protein alternating with lipids. To be more specific, we have lecithin, cerebroside and cholesterol layers which will be alternating in between the concentric layers of protein. So myelin sheath is not at all a continuous sheath. At specific interval, this myelin sheath will be absent. The point at which the myelin sheath is going to be absent is called as node of Ranvier. So as I said before, the myelin sheath is going to be a thick lipoprotein sheath. So the process of formation of thick lipoprotein sheath is called as myelinogenesis. So this myelinogenesis is formed by a special type of cells which are called as Schwann cells. So these Schwann cells are present in the neurolemma. What is neurolemma? Neurolemma is going to be a covering sheath which covers the axis cylinder. So the neurolemma is a thin membrane which surrounds the axis cylinder. We have discussed this in the previous slides also. Here we have a picture representation on both myelinated nerve fiber and non-myelinated nerve fiber. So as we have discussed before, there is going to be a central core of cytoplasm which is called as axoplasm. So this axoplasm will be surrounded by a layer which is called as axolemma. So after axolemma, we have a layer of myelin sheath in case of a myelinated nerve fiber. And again, this myelin sheath will be surrounded by a layer of neurolemma. So this happens in case of a myelinated nerve fiber. So first thing will be the central core of cytoplasm, that is the axoplasm, surrounded by axolemma, which is again surrounded by the myelin sheath, then followed by the neurolemma. Again, when we come to discuss about this non-myelinated nerve fiber, same thing will be there. There will be a central core of cytoplasm, that is the axoplasm, surrounded by the layer which is called as axolemma. But here we will not have a myelin sheath. So this axolemma will be directly covered by the neurolemma. So you can look back to this slide. In a non-myelinated nerve fiber, the neurolemma surrounds the axolemma. In a myelinated nerve fiber, it will be covering the myelin sheath. So in non-myelinated nerve fiber, the major function of the neurolemma is to cover the membrane. In a non-myelinated nerve fiber, this neurolemma will be serving as a covering membrane. Whereas in myelinated nerve fiber, the neurolemma is very much essential for the formation of myelin sheath. So it is a very important point to be noted. As I have discussed before, Schwann cells are very much essential in the process of myelin sheath formation. So in case of central nervous system, the Schwann cells are absent. So what will happen? We have a neuroglial cell which is called as oligodendroglia. And these oligodendroglia are responsible for the process of myelinogenesis in the central nervous system. So we can consider in this way. So the Schwann cells are responsible for the process of myelinogenesis in peripheral nervous system, whereas this oligodendrocytes, the neuroglial cells, are responsible for the formation of myelinogenesis in the central nervous system. So in the peripheral nervous system, we have Schwann cells, and in the central nervous system, we have oligodendrocytes. Then we will discuss about the functions of myelin sheath. So the two major function of myelin sheath is going to be faster conduction, and the other one is going to be the insulating capacity. Yes, the myelin sheath has the ability to conduct action potential in a very fast manner. I think we have to talk about the saltatory conduction when we are talking about the faster conduction. Yes, as I have discussed before, myelin sheath is not a continuous structure. It is going to be absent at a regular interval. So at that regular interval, there is going to be a node which is called as node of Ranvier. So because of the presence of this node of Ranvier, the action potentials will be keep on jumping from one node of Ranvier to the other node of Ranvier. Because of this, the conduction of action potential is going to be more in case of myelinated nerve fiber. Next, to talk about insulating capacity, the myelin sheath has a higher quality of insulating power. So this quality restricts the action potential to be transferred within the cell rather than sharing the action potential to the neighboring cells. So next we will discuss about the axon terminal. The axon terminal is going to be a specialized organ. This specialized organ will be present at the end of the axon. So at the end they will be containing a finger-like projections, and this axon terminal is very much essential in releasing neurotransmitter, and they will be releasing neurotransmitter in a close proximity, but they will not touch other nerve fibers or muscle. So the major function of this axon terminal is to conduct action potential. So here you have a picture representation of the axon terminal. Here you have the cell body, the axon along the myelin sheath, and you can see the axon terminal, which is a finger-like projection which is getting connected to the other neighboring neurons in order to send the signals for processing. So now we'll have a quick recap on today's session. So in the introduction, we discussed that the nervous system is mainly composed of two major cells. The one is going to be the neuron and the other one is going to be the neuroglial cells. So first we started with the neuron. So neurons are the basic structural and functional unit of the nervous system, which are the true functional cells that are responsible for conducting action potential. Then we started with the neuroglial cells. These neuroglial cells are the supporting cells which are non-conductible cells. Then we discussed about the structure of neuron. In that we started with dendrite. So dendrite is going to be the extensive branching which are tapering distally. So they form a dendritic tree. As they are tapering distally, they will be reducing in their diameter. Then we discussed about the soma. The soma is also known as cell body. So the cell body will consist of nucleus and other essential organelles. To be more specific, we had nucleus granules. This nucleus granule is very much responsible for the process of protein synthesis. Then we started with axon hillock. So axon hillock is going to be a triangular conical shaped structure which will separate the cell body from the axon. So this will be extending as the axon further. And then we spoke about axon. Axon is going to be a tubular structure which will have a central core of cytoplasm which is called as axoplasm, which is surrounded by a layer of axolemma, and again that layer is surrounded by the myelin sheath followed by the neurolemma. So again we discussed about myelin sheath. Myelin sheath is going to be a thick lipoprotein sheath which is covering the axon. So it is formed by the concentric layers of proteins with the alternating layers of lipid. So this myelin sheath is not going to be a continuous structure throughout. Rather, they are going to be absent at a regular specific intervals. So the point at where this myelin sheath are absent, that point is called as node of Ranvier. At this node of Ranvier, the action potentials keep jumping from one node to the other. This will result in the faster conduction that is called as saltatory conduction. Again, we discussed about the axon terminal. So axon terminal is going to be the end portion of the axon which are in the form of finger-like projection. So this structure is very much essential in transmitting action potential from one neuron to the other or from one neuron to the muscle. So this axon terminal is responsible for sending signals for further processing. So with this we have come to the end of today's session. Thank you.